Good afternoon, students. Jai Hind. Today we will be discussing the evidences of evolution. The learning objectives. At the end of this discussion, the students will become aware of the various evidences of evolution. Students will know about fossils and connecting links. They will know the difference between homologous and analogous organs. They will know the difference between convergent and divergent evolution. Students will also learn how human DNA is only 1.8% different from that of chimpanzee. Evidences of evolution. Now, if we have to accept anything, then we require an evidence for that. So, for evolution also, there are evidences from various fields, namely paleontological evidence, evidence from the comparative anatomy and morphology, biochemical evidence, embryological evidence, and biogeographical evidence. So first of all, paleontological evidence. Paleontological evidence which involves fossils. And what are fossils? Fossils represent the plants and animals that lived millions of years ago and are now extinct. Different aged rock sediments, they contain fossils of different life forms which probably died during the formation of the particular sediment. Here in the slide you can see some of the fossils and have a look at the prehistoric life. Then we have certain evidences from comparative anatomy and morphology. It shows the evidences of the similarities and differences between living forms of today and that of prehistoric times. Some of the examples of the comparative anatomy and morphology are connecting links, homologous organs, analogous organs, and adaptive radiation. Now let us look at these one by one. So what is this? Can anyone identify? It is a connecting link of Archaeopteryx, which is a transitional form between birds and reptiles. And birds are said to be glorified reptiles. So this is not the first one, but this is one complete image of the fossil. Then homologous organs. Homologous organs, homo means same. So whale, bats, cheetah and human, they share similarities in the pattern of bones of forelimbs. The forelimbs, they perform different functions in these animals, but they have similar anatomical structure. That is, all of them, they have the various bones like humerus, radius, ulna, carpals, metacarpals and phalanges. Hence, in these animals, the same structure developed along different directions due to adaptation to different needs. So, because of different needs, the organs, that is the bones, were the same, but externally they were different, and this presence of homologous organs, it shows divergent evolution. Or we can say homology indicates common ancestry. That is, from a common ancestor, all these animals have descended or developed. Other examples of homologous organs, they are vertebrate hearts and brains. Then in case of plants, thorn of bougainvillea and tendrils present in cucurbit, they also represent homology. So you can see homologous structures, they have common evolutionary origin, that is common ancestor, but can look different and have different functions. So pentadactyl limb. So pentadactyl means 5-digited limb, which you can see in bird, in bat, horse and human. They have all the same bones, arrangement is same. In all these animals, humerus is present, radius you can see, ulna, carpals, metacarpals and phalanges. Then we have the evidence from analogous organs. Analogous organs, that is wings of butterfly and birds. That is, in appearance and function they are same. They are anatomically dissimilar, but they perform similar function. So we call such organs as analogous structures, and they result in convergent evolution. That is, for a similar function, different anatomical structures, they are performing same function. Their appearance becomes same. Other examples are like eye of octopus and eye of mammals, flippers of penguins and dolphins, then sweet potato, which is a root modification, and potato, which is a stem modification, are other examples of analogous organs. So here in this figure we can see some of the analogous structures in case of plants as well as in animals. Then we come across adaptive radiation. Adaptive means the structures they are modifying for a particular habitat. So a very good example of this is that of Darwin's finches. In Galapagos Islands, Darwin observed small black birds which were later known as Darwin's finches. He realized that there were many varieties of finches in the same island. All the varieties he came across evolved on the island itself. From the original seed-eating features, many other forms with altered beaks arose, enabling them to become insectivorous and vegetarian finches. So this process of evolution of different species in a given geographical area, starting from a point and literally radiating to other areas of geography, is called adaptive radiation. So you can see some of the Darwin finches. Adaptive radiation, Australian marsupials. A number of marsupials, each different from other, evolved from an ancestral stock, but all within the Australian island continent. Then there are placental animals also found in Australia, also exhibit adaptive radiation in evolving into varieties of such placental mammals, each of which appears to be similar to a corresponding marsupial. So here we can see the marsupial radiation that is found in Australia. And in this slide we can see placental mammals and Australian marsupials. When more than one adaptive radiation, we are observing adaptive radiation in placental mammals as well as Australian marsupials, and it is occurring in the same place, Australia. So when more than one adaptive radiation appeared to have occurred in an isolated geographical area, we can also call this as convergent evolution. The structures, they are very much similar because of their similar needs. So the evolution is convergent evolution. Then biochemical evidence. We also get evidence of evolution from the field of biochemistry. It is observed that all living organisms use DNA and RNA as genetic material. Human DNA, it is just 1.8% different from that of chimpanzee. All organisms use 20 amino acids to make their proteins. And there is similarity in structure of actin and tubulin protein, which proves a common ancestry. And the genetic code, this is common to all life forms found on the earth. So this is the evidence from the field of biochemistry. Then from the field of embryology, again we can get some evidence. They are the embryological evidence. Ernst Haeckel had proposed a theory that is ontogeny recapitulates phylogeny. This means an organism in the course of development, that is in the embryonic stage, it goes through more or less the same successive stages as did the species in its evolutionary development. So ontogeny, that is during embryonic development, and phylogeny, that is evolutionary development. So ontogeny is recapitulating phylogeny. The embryos of all vertebrates including human develop a row of vestigial gill slits just behind the head, but it is a functional organ only in case of fish. It is not found in other adult vertebrates. Now the theory of ontogeny recapitulates phylogeny. This was disproved later on by careful study performed by Carl Ernst von Baer. He noted that the embryos never pass through the adult stages of other animals, though they are similar in appearance. So we can have a look at this slide. Embryos of vertebrates, they look similar. Almost all mammals, as you can see, they have got 7 cervical or neck vertebra and the gill arches are present. And ontogeny recapitulates phylogeny as given by Ernst Haeckel. So here in the slide you can have a look at the embryonic stage of various animals, the embryos, but Carl von Baer, he said these embryos, they never pass through the adult stages of the animals. Then we have evidence from the field of biogeography as well. The study of the distribution of living things over the earth. Now the question, why some species we find they are absent from environments that would suit them? Similarly, why are there closer relationships between species that are geographically near each other between the species that inhabit similar environments? In Asia, we find many types of placental animals and very few marsupials. But in Australia, we find mostly marsupials, kangaroo, and very few placental animals. So polar bears, they always, you will find them in Arctic, you will never find them in Antarctic, but penguins, you will only find them in Antarctic and never in Arctic region. So by looking at these various evidences, we can conclude that evolution has taken place and there was a common ancestor. Due to changed conditions, adaptations took place and modification with descent was observed. So thank you.